highlights. He's the Senior Research Fellow in Ecology here at Harvard Forest. He did his undergraduate at Yale, and then a PhD in ecology at Brown University. Postdoc work at the Organization for Tropical Studies, OTS, in Cornell, and then was a professor at Mount Holyoke College for many years before coming to Harvard Forest around 2001 and becoming a senior research fellow in ecology. He's published widely in sort of ecological theory, ecological studies, natural history, published numerous books that many of us are familiar with. The Primer of Ecological Statistics is sort of a classic now amongst grad students and ecologists. The Field Guide to Ants of New England. A great new book is out on carnivorous plants, as well as dozens and dozens of research papers. So very well-known ecologist, and we're excited to have him here this evening to sort of host us and present to the group, talking about things fall apart, land use history, non-native insects, climatic change, and the decline of the forest foundation species. So, thank you for hosting, Aaron.
Thank you very much. It's actually really fun to have the opportunity to give a science talk where I work. I was telling people over dinner in the lab, this is 3 talks in 10 days. I gave a talk at the Atlanta Botanical Garden. Actually, it's like 4 talks in 3 weeks. I was at Yale just a couple weeks ago at the Yale Forest Forum talking about hemlock and loss of foundation species. I was at the Atlanta Botanical Garden talking about carnivorous plants, and I was at Lawrence University in Wisconsin on Monday, and I was supposed to talk, what was I supposed to talk about? I don't remember what I was supposed to talk about. I think I was actually supposed to talk about, I was talking about ants and climate change, because I do a lot of work with ants and things with climate change. And I was giving a talk on Monday at 3 in the afternoon, and I woke up Monday morning and IPCC had released its report about the world going to hell in another handbasket this week, and I thought, you know, I really need, we're talking about ants and climate change. Why am I talking about ants and climate change when IPCC is reporting out? So I tacked on 10 minutes about Wisconsin at the beginning and what they were likely to see. And tonight I'm going to talk a little bit more, again, locally, because we're here and it's New England and we care about hemlocks. And so I just wanted to have the opportunity to talk to you all about some of the work that we do here at Harvard Forest, focusing on our major forest species and on how our forests are changing. And just to give you context about where you are, I know you drove up here, you've spent the whole day here, but fun facts and figures about Harvard Forest that are useful to know. We are in the least densely populated, most heavily forested section of Massachusetts here in central Mass. And you know, I go around the world, I work all over the world now, and when I give talks in China and I say, okay, so this is a region where Harvard Forest is, this is the size of Beijing, right? And Beijing has 35 million people in it, and we have 30,000. And it just blows them out of, it's just an incredible thing. And we have many Chinese colleagues come here and work with us, and we say, look, you know, we're not in Boston. Got that, we're not in Boston. We're a third world country here in the United States. There's no mass transit out here. There's nowhere to, you know, go grocery shopping. No one from overseas can ever imagine this, right? And then they get here and like, how do I get to the grocery store? Many of you, I hope, know we're here right at the northeast corner of the Quabbin. You all know that the Quabbin is Boston's water supply. You all know, of course, that it's an untreated water supply. It's a filtered water supply, but it's untreated. It's one of the cleanest water supplies in the world. And in part, this is because this whole region has been allowed to regrow. When Harvard Forest was established, as I mentioned when we were out on our walk, this landscape was 20% forest cover. In fact, I have, this was unplanned, this was forest cover in the North Quabbin in 1830.
It's really hard to see. Can you take it over? Take it sideways out of the light. There you go.
1830, forest cover in the North Quabbin in that box, right? The black is forest cover in 1830. at the height of agriculture. And this is forest cover 150 years later in this region, right? For me, working here in New England, I am not a native New Englander, but I've lived in New England for more than 40 years now. I love living in New England. It's the greatest restoration story on the planet. The Europeans came, they completely deforested the landscape, then they discovered industry to the east and good soils to the west. They threw the keys on the table and left. And we got magnificent forests here. We got functioning ecological systems. It is such a great place to work. But we didn't do anything. We didn't go out there planting trees. We didn't do anything. It comes back. It's really amazing. We spend a lot of time here at Harvard Forest. I mentioned out in the woods, we're an LTER site. We're a NEON site for the Northeast US. We really focus on the intersection between people and nature. And we don't really, all the work that we do out here, people is nature. We don't make this distinction in a big way. We work in sort of basic ecology, applied ecology, serving out decision support science to policymakers and decision makers throughout New England, the United States, and the world. And it's a great place to work. I've been here since 2001. My first day at the office was September 11th, 2001. So fun fact. Right? Today I'm going to talk about decline of, and I'm going to talk about forest change viewed through the lens of how forests change when we lose what ecologists call foundation species. This is not a hemlock, although we like to call hemlocks the redwoods of the East. And the idea of a foundation species is a species that exerts disproportionate control on all the biological processes and the abiotic processes that function and drive the system that they structure and define. They were originally identified and categorized for marine systems back in the 1970s in Antarctica. And I guess we have the dubious distinction of introducing the concept to the terrestrial realm. Back in 2005, we wrote a paper that came out in Frontiers in Ecology and the Environment, and we said, you know, this is a useful concept. It's changing our forested ecosystems here. We should think about this. And myself and a whole bunch of other people here at Harvard Forest and elsewhere around the world have been working on this assiduously ever since.
How is it different than keystone species?
Well, how is it different than keystone species? Can I punt on that for just a minute? Right? The short version is keystone species are top predators or high-level predators that control intermediate levels of diversity by eating dominant competitors and exert top-down effects, whereas foundation species are bottom-up effects. And we can spend another hour on that. Right? So we'll deal with that later. But there are biological aspects to foundation species. And as we saw walking around in the woods today, through Hemlock Hospice, there are cultural aspects as well. And one of the ways that we identify foundation species that we don't identify keystone species as, at least keystone species sensu stricto, the way Bob Payne defined them as opposed to the way Buzz Holling defined them, is they name the system. So we think about hemlock forests, or we think about redwood forests, or we think about mangrove forests. We don't actually think about starfish shores, where, you know, Physaster is your classic keystone species. Foundation species like eastern hemlock have tremendous cultural resonance, right? Everybody in New England, certainly in the East, you know, we have a mental image of hemlock forests as something profoundly different from most other kinds of forests here. Our poets, Frost, Dickinson, Updike, the list is really, really long, write poetry about hemlock forests. We don't write poems about Physaster. I am going to assert tonight, that's another talk, how we got to that assertion, that eastern hemlock is a foundation species in our forest. Now, we talk about foundation species and their existence as a hypothesis to be tested, that you actually have to go out and show that this species really does disproportionately affect ecosystem processes. It really does disproportionately affect biodiversity. I'll give you a couple of examples, but experimentally and observationally, we've been working on that here explicitly for about 20 years, trying to really demonstrate this and nail this down relative to other species in the system. Harvard Forest has a more than 100-year history. The last photograph I showed you, our book on hemlock that came out a couple of years ago, covers the history of hemlock research here at Harvard Forest. We couldn't do, for example, the same kind of book on oak or chestnut here at the Harvard Forest. They're important species, they do a lot, but the resonance of hemlock, even here at Harvard Forest, is disproportionate to sort of everything else we do. For the applied foresters in the room, hemlock, eastern hemlock, is a major component of more named forest ecosystem types in the U.S. Forest Service classification than any other forest tree in North America. Right? So even though, you know, here's its range, it's about, what, 6 million hectares, 6 million square kilometers, something like that, over the eastern North America. This is a major component of our forests. Here are a couple of examples of some of the things that hemlock does in our system. This one, we saw the eddy covariance towers when we were out in the middle of the installations. These are 3 traces of carbon flux, soil temperature, and soil moisture in a hardwood stand in the environmental monitoring system, the eddy flux tower that's been there since 1991. In blue, a young hemlock stand. We used to have another eddy covariance tower. Those of you who walked to the Black Gum Swamp would have later on that path walked by the other eddy covariance tower, but you probably didn't see it. And then we had the one standing there where we were in the middle of the forest, and that's the old hemlock stand in green. And this is over a couple of years. This is just a 5-year trace, 6-year trace from 1995 to 2000 that importantly spanned a very dry year in 1999, just as an illustration. And so the carbon flux in 1999 for the hardwood and the very young hemlock stand with a lot of other things in it is really strongly affected by drought, not surprisingly. But the hemlock is the old hemlock stand, the green one, is trucking along just fine. It just keeps modulating and maintaining the ecosystem processes as measured here as carbon flux throughout this in a very sort of smooth and even consistent way. Soil temperature is similarly very nicely modulated, more evenly in hemlock stands than in hardwood stands, and similarly soil moisture is much less responsive. And as an interesting side note of this, as hemlock has been declining throughout the east, hemlock is strongly, in many parts of its range, strongly associated with riparian corridors, with streams and lakes. Streams and rivers. And when hemlock declines, the rivers become much flashier because you don't have that sort of bridge in mud season or in late fall when hemlock is still able to photosynthesize and transpire, and it keeps those streams much calmer. And so as it's replaced by deciduous stands, you get this much more flashiness to the streams. And so one of my questions about sort of historic New England is when Europeans got here and they took a lot of the hemlock down, it was harvested extensively for tan bark, it was almost completely eliminated in the White Mountains for the tanning industry in the 1800s. What did that do to the hydrology of the streams? In principle, it should have made them much flashier, and maybe that could have contributed actually to the development of hydropower along those streams, because now you have to figure out how to dam those streams and manage the water level for mills and things like that. I have no way to test that. It's just an interesting thought experiment. Hemlock is also really interesting in terms of what it does to local fauna. When I talk to my students, and particularly the undergraduates come in the summertime, and they say, oh, hemlock's a foundation species. That means there's much more biodiversity in hemlock forest than there is in hardwood forest. And I said, no, all I said was that hemlock controls the biodiversity. It turns out that for some taxa, ants, because I work a lot with ants, being my example here, you actually have far fewer ants in hemlock forest than you do in hardwood forest. And you in fact lose two genera, Formica and Lasius, are almost completely absent in our hemlock forests, right? And this could be because of temperature, it could be because of light, there are all sorts of things that we've looked at, but it's really extraordinarily striking. On the other hand, spider diversity is a couple orders of magnitude higher in hemlock forest than it is in hardwood forest. There are a few interesting birds that are more focused on hemlock forests than elsewhere. By and large, though, hemlock forests up here are not where you go for biodiversity. On the other hand, when you take hemlock out of the system, your landscape diversity is much more homogeneous, right? You don't have this patchy structure. Hemlock is declining. We talked about this out in the woods. These are just some illustrations of the adelgid. The adelgid is this small mealybug-like insect here. Here it is in the fluffy wool stage. This is a shot from down in southern Connecticut some years back as hemlock declines. This is why we close our trails, because hemlocks fall apart. And this is what people do. It turns out that there is, of course, as we talked about, out in the woods, there's some economic value to hemlock, either as saw logs for post and beam construction, if they're particularly good, or for biomass. We have now lost in New England, in southern New England, about the same amount of hemlock to preemptive salvage logging as we have to the adelgid. Because once the adelgid gets it and it gets punky and it falls apart, it's not worth anything. So the logging prescription is to go cut it before the adelgid gets there. Now, this is not unique. We did it to chestnut, right? And now we did it to elm. We've done it to hemlock, and now we're doing it to ash. All right, so we got the emerald ash borer on the way. The standard forester prescription now in New England of what to do with your forest, if you've got ash on it, is to cut it, all of it. Mill it, billet it, put it in storage. What's ash used for?
Baseball.
Baseball bats.
Baskets.
And the Major League Baseball doesn't use aluminum, right? And that is the major market for ash. It's baseball bats, and it's all being salvaged. And the ash bore's here, right? But it's not everywhere. But it's all getting cut, right? So there's this intersection here between people bringing insects and insects doing what they do and people doing what they do, and the end result is a very different kind of forest system. This is the data from the Forest Service from 2015 of where the adelgid was at the end of 2015. I've been in the process of putting together this week a review paper on the impact of the adelgid on our forests in New England, and I contacted the Forest Service and I said, so do you have an updated map? Because the last one you can get on their website is 2015, and they said, no, we're not tracking it anymore.
What?
I just work here, right? And one of my former students is now working in West Virginia, and she sent me a note the other day. She's like right here. And she said, we got it. And I said, well, you can call the Forest Service and report it. I don't know what they're going to do with it. And of course, it's only the U.S., right? Even though hemlock's up here, they don't report anything in Canada. Supposedly, Michigan is trying to eradicate, but it's all there, and of course it's all through the Adirondacks now, and it's up into central Maine. But as far as I know, the Forest Service is no longer tracking. And the previous one. This just illustrates, Harvard Forest has been studying hemlock, and some of the data I'll be talking about comes from permanent plots that we established along a transect going from Long Island Sound up to here. We set up this transect in 1992 to here, and then in 2000 we extended it up to the Massachusetts border, and in 2008 we extended it into Vermont and New Hampshire. And so we have hundreds of permanent plots scattered across this transect, and we've mapped using remote sensing imagery all the hemlock all the hemlock in that entire belt. And we've been tracking the process of hemlock decline and the adelgid movement in this large belt transect for going on for almost 30 years now. And this is what happens. This is basically what happens. When hemlock declines, you get a flood of light to the forest floor. Hemlock doesn't seed bank. The seeds last about 1 to 2 years, and so it's really not a seed banking species, but birch seed banks very well. And within about 10 years, this is a photo of 10 years after this one, you've got a very nice thicket of black birch. And many of our regions, some mix of red maple, but for here in southern New England, this is about the general progression that we see. So since I started working here at Harvard Forest, among other projects, I've been working on what happens to these systems when you take hemlock, a foundation species, out of this system. I'm particularly interested in how ecological systems reorganize themselves following any kinds of disturbance, and loss of hemlock is one of these. How is this related to the hemlock, the biology of hemlock itself, the biology of the adelgid? Where is it going, right? How does what we do in this system, how does what the adelgid do in this system change the trajectories, change the endpoints? And is this completely novel? We spend a lot of time and energy in ecology these days talking about novel ecosystems. Can we learn anything from the past as well? This is a project, pieces of which I run, pieces of which I collaborate on, pieces of which other people run and do. It's a big, large focus, a big chunk of our long-term ecological research program here, involving literally dozens and dozens of researchers, from undergraduates to senior researchers, looking at things ranging retrospectively through pollen analysis and fossil analysis to spatial patterns of insect spread and human activities, down to the micro level of physiology, what the trees are doing, what the insects are doing, what's going on at the soil level, and then scaling this back up to macro levels to try and understand where these systems are going. Most of the work that I'll talk about today, of course, is being done here at Harvard Forest. The red outlines here, the green is our, and yellow are our different kinds of hemlock stands. Some of the experimental work I'll be talking about is at this plot, this tract here, the 350-acre Sims tract in I guess southern Petersham. Petersham does extend all the way down in this tongue. We're located there in central Mass. I'll be talking about some pollen cores that we've taken at 4 other locations in the state, shown there with the triangles, and I'll be talking about data from the Connecticut portion of the transect as well. So all of those will figure into some of the data and results that I'll be showing you. The pollen cores are quite interesting. I'll hone in on those 4 ponds in just a second, but just to give you a sense, as we were again talking about out in the woods, of the dynamic nature of our forests. We take pollen cores all over New England. We have one of the densest assemblages of pollen records anywhere in the world. Here in Massachusetts, Harvard Forest researchers have sampled upwards of 50 different sites in Massachusetts alone. And this is a standard pollen diagram that comes out of these things. You've got from glacial recession here about 14,000 years ago to the present going up. You've got all the taxa going along the top, and then you've got the percentage of their representation in the pollen core. So after the glaciers went away, we had spruce forests. We had spruce forests for about 4,000 years, then it got warmer, and then we had hemlock forests. And we had some pine in there, and then pine came back. And then we got some birch, and these other taxa doing their thing. Black gum was really big ones. And then we've got that period of time that all of us romanticize and remember, when we had farms and we had cows and we had agriculture in New England. That's this little tiny slice right here. When we've got grass and we've got ragweed and a little bit of sedge and chestnut. Because chestnut's actually an early successional species too, and it did really, really well after settlement and land clearing. And so that's where you get those nice big chestnuts up here. And then they're gone too. So we have this long dynamism, and if we just simply look at oak and maple alone between the colonial era and the modern era from these pollen cores, you can see this sort of wholesale shift in our landscape from a really strong oak-dominated landscape to a much more mixed maple oak-dominated landscape. Oak was heavily used, and it's slower to come back. If we look in more detail at these four ponds that we sampled, including one up here in Royalston, Little Pond, we look at these are representative of all these others. In fact, they're representative of most pollen cores that we would take throughout eastern North America. And the key feature here is about 5,700 years ago, this is hemlock here, about 5,700 to 5,400 years ago, hemlock, which was really, really dominant everywhere, all of a sudden crashes. And it crashes almost synchronously within the resolution of fossil pollen, which is about 50 to 100 years. It crashes synchronously across eastern North America. And Margaret Davis, who worked on part of this for her dissertation work, hypothesized that it was caused, this hemlock decline, range-wide hemlock decline, was caused by the insect looper, the hemlock looper, a native geometrid moth, and that this was an outbreak throughout its range and it caused this decline. Subsequent work has shown that that's a bit harder to support. We actually don't find very much fossil evidence for the looper in any of these cores, and you'd think you'd find some, but you don't find a lot. But at the same time that this was happening, we have drying events as it's getting much drier, it's getting much warmer in the region, and so now we think that this is more complicated in terms of thinking about climatic change as well as insects, but it was clearly a range-wide effect, and it took about 1,000 years for hemlock to regain its former dominance. And so when we think about loss of major species like hemlock, perhaps like chestnut, that we with our 70, 80, 90-year lifespans get all bent out of shape about, it's kind of useful to keep in mind that, you know, the trees are working on 1,000-year lifespans. You know, this is 3-4 generations for a hemlock, right? It's 20 generations for us, 50 generations for us. That time scale is really important for us to keep in mind as we think about how these forests change. Of course, we're surrounded by these dioramas, and I hope that all of you who've had a chance to watch, I mean, this is such a pleasure to be able to talk about the dioramas, and this one is like right in the corner, right? These dioramas were put in the 1930s, all right, and they showed a history of land use in New England, going from the primeval forest and land use to abandonment and succession, and then the middle one, when the museum was actually built. So if any of you haven't been here in the last 2 years when the museum was renovated, you would remember that there used to be walls here, and that one there was a post that blocked that one, because you were supposed to be able to walk in the door and see that, the Tom Swamp diorama, which is a double diorama, as sort of Richard Fisher, the first director of the forest, that was his favorite view. All right, so that gets the double line. And then on the other side, our whole series of dioramas about how to manage forests. And all of this was done in the 1930s, right before the 1938 hurricane. Right? But it's important to remember that this is our forest. This is Harvard Forest 150 years ago. Right? And so even 150 years ago, our landscape was completely different. After the land was abandoned, after people moved to industry in the east and agriculture in the Midwest where the soils were better, the forest began to recover, right? And this is data from the Forest Inventory Analysis Program. The Forest Service has plots all over the world, all over the country, and they go sample them. And we got the data here through early 2000 for the 4 southern New England states. They began the FIA program in the mid-1940s, and so we can see the change in growing stock in millions of cubic meters of hemlock just in each of these states. And it's just gone, it had been going steady up from the 1950s. It stopped going steady up in Connecticut in the 80s after the adelgid got there. The adelgid first got into Massachusetts in the late 80s. Massachusetts is now leveling off. Vermont and New Hampshire are still on a pretty much upward trajectory with hemlock, as with everything else. I mean, our growing stock in our forest, it continues to be quite good in New England, although there is continued harvesting and development pressure. But because hemlock is not a particularly economically important species, other than pulp and things that you might get a little bit out of, it hasn't been heavily logged since settlement. And then the adelgid showed up in the 1980s. We talked out in the woods about how the adelgid is limited in its northward progression by temperature. And so the minimum temperature of minus 25 degrees on a cold winter night. And when you stop getting that, the adelgid moves in. And so the last time we had one of those in southern Connecticut was in about 1984, and the adelgid was well established in 1985. In Storrs, Connecticut, at Yukon, it's a little bit later, and the adelgid is there. And at Harvard Forest, our last minus 25 degree night when I put this figure together was in 1998. We did go down to minus 29 last year for one night. It had no effect on the adelgid because the adelgid has been subject to very strong selection, right? And it's just the resistance to that continues to decline. To cold, the resistance to cold continues to increase. It no longer has the declining effect on it. I would point out, you know, we talk about how cold it was last winter. It just wasn't that cold, right? I mean, historically, you know, Harvard Forest, you know, would go down well below, you know, to minus 30 and below. And, you know, that's just inconceivable to us now. So, you know, minus 23. And when I lived in Royalston, my card tells me what temperature it is. And I remember a couple of days, minus 20, and I'd be like, wow, this is brutal, right? But it's just not that cold. The adelgid really does track temperature mostly, right? So here's the data for the adelgid spread and where it went to first and how quickly it got there. So it started here, then it showed up here, and we got the big explosion here, and then it went down south, right? Which is really not exactly what climate would predict. Climate would predict it would go this way first and then up. And when we did some sort of basic spatial modeling using climate, it sort of says, well, you go this way and then you go this way. There, of course, is more hemlock in the north. But the basic models fitting with climate actually don't do a particularly good job of predicting the adelgid. It underpredicts when it gets to the northeast, and it overpredicts when it gets to the southeast. And it turns out that people have a lot to do with this. And so we ran simulations. We ran, you know, thousands of simulations of where the adelgid ought to go, knowing about its dispersal, knowing about climate, knowing about how many hemlock trees there are. And all of these different lines are the different trajectories that each run of the model predicted. And then the solid circle is where the climate model would project it would go over time. So that's sort of the midpoint of the model, as it were. And so the model says, yeah, you know, we ought to be here until it gets warmer, and then it starts moving north. This is what the adelgid actually did in terms of the center of mass of the adelgid distribution. First it moved up here, and then it moved back, right? And I show this for two reasons. The first is, of course, we know from historical records that people moved it here. So we know that people transported it from Virginia to Pennsylvania to Connecticut. But the other thing is, and this is, you know, thinking about the IPCC report that just came out, we put a great deal of faith in models, right? We got these huge models, we run them all the time. I do a lot of modeling, my colleagues do a lot of modeling. We believe this. We believe the ensemble means. Nature doesn't care about the ensemble means. And more importantly, what we actually get is not the ensemble mean. We get one trace. It could be on the mean, it could be somewhere else, right? We could get a hurricane that all the models say it's going to hit as a tropical storm. Oh no, it's going to hit as a Category 1. Whoa, it's almost a Category 5. We didn't predict that. Well, but that's, your model actually had that. It just happened to be 4 standard deviations away, so it didn't show up on the ensemble. Right? We put a lot of faith in models, but it's not always the best place. When we think about what's going to happen to our forests, we have models. We have hypotheses. We can quantify these. We might say that when a foundation species such as hemlock is present, it's going to really affect the biodiversity in the system. It's going to really affect the ecosystem processes in the system. When you take it out, those heavy effects disappear and you have much more direct effects of predators that are driving the system, or you have microbial processes that are much more important. And this is a model, this is a conceptual model that we can then test experimentally or with other kinds of data. So we did this, right? We took a hemlock forest, this is our hemlock forest, and we applied 4 different treatments to it. Back in 2003, we identified 8 plots in the Sims tract. We've got 3 that are down in this valley here. How many of you went out to the Soapstone Ridge today? Right, so that's up here. It's just above this, right? And then 4 plots on the ridge up above this. So we got 2 blocks. By ecosystem standards, this is a replicated experiment. We got 2. Better than 1. Each of these plots is a hectare in size, 2.5 acres. These 2 sets of 3 were each initially completely dominated by hemlock. More than 80% of the canopy and basal area was hemlock, and then we had 2 young hardwood stands, which were where did we expect this forest to go into the future. So we have 2 blocks. We have different starting conditions, hemlock and hardwood, and then in the the hemlock plots, we have a control plot. We have a plot where we killed all the hemlocks standing in place, so that we were pretending we were adelgids, and we girdled them with chainsaws and knives, and watched them decline. And then we had a plot where we pretended we were commercial loggers, and we went in and we took out all the hemlocks bigger than 8 inches DBH, or 4 inches, so 10 centimeters DBH. And then we were real loggers. We got a crew here. They love going out into the forest to cut trees. This building, as our whole central campus is heated by wood that we harvest right here at Harvard Forest. We burn 80 cords of wood a winter to heat about 100,000 square feet. So we do a lot of cutting of wood. The barns that you saw as we walked by, we built. The maintenance garage was built from this plot. But as I've said repeatedly, hemlock is not particularly valuable, and so what a commercial operation will do is they'll go and say, okay, I'll take out that hemlock, but you know, hemlock is not going to pay for that cut. But if I take that white pine and that red oak and that white oak and yeah, we'll take a couple of these others, now you got some good saw timber, that'll pay for your cut and you'll make some money. So we did the same thing. We took out the merchantable white pine and the merchantable red oak that were big, and we left a couple of seed trees, but otherwise basically did a clear cut of the hemlock down to 10 centimeters DBH. We took 2 years of data on this before we did the treatment, so 2003, 2004, and then we applied the treatments in 2005. The idea here, when we did this in 2005, the adelgid was not here yet. And so the idea was that our girdled plots were going to tell us something about how a forest, a hemlock forest, used to be hemlock forest, would change subject only to physical decline of the tree, and that eventually the adelgid would get into our control plots and they would kill those trees, and those would let us know what's going on when the trees are both declining physically and have an insect in it. So initially, our initial comparison was between our control plots versus our girdled plots and our log plots, and also to see the difference there. But now that the adelgid got here starting in 2009, our interest really is the comparison between the control plot and the girdled plot to try and tease apart the actual effect of the insect that is different from the effect just of the physical decline of the tree. All right, so this experiment is actually set up to ask multiple levels of these kinds of questions. And then our hardwood plots are our sort of future reference controls. And so we put this in, and we put this in at the Sims tract, and of course this is not virgin forest, this is not primary hemlock forest, this has really interesting land use and people history. I spent a year in the archives reconstructing the land use history. We published this article in Rodora a couple of years back. This whole tract, the Sims tract where our plots are located, was originally at settlement and division in 2 farms. The north part basically stayed in one big block for the last 200 and some years, 250 years. The bottom part was subdivided and recreated and subdivided and recreated. It was like real estate flipping, like you could not believe. And in fact, the appendix in the Rodora article has all the deed records going through all this. It's all been thinned, it's been cut, it was beat up during the 1938 hurricane. There's a sugar operation over here, there's a historic cider mill. I mean, this whole area was worked. Right? At the same time, the population was changing, the climate has been changing, we've had episodic droughts and insects and whatever. Through all of this, we've got hemlock in different places and slowly growing and changing the way this forest works. You can really see the development of this forest, particularly since the 1938 hurricane. Let's get this out of the way here. So here's some plots here. These are our hardwood plots at the bottom and our hemlock plots at the top before we started the experiment. So we have distributions of diameters and ages of the trees, and most of the trees are relatively small and relatively young. The oldest tree that we've got is about 140 years old in this plot, about 140-year-olds here. We've got a good relationship between size and diameter in the valley, and not such a good relationship on the ridge, which was thinned in the 1920s. The young hardwood stands really are very young. We have one relatively old white pine in one of our plots. So we actually know we did complete chronological reconstruction of the centers of these plots, the center 900 square meters, just like you did in your tree ring workshop. We did tree cores of, I don't know, 700-800 trees to figure out their history. In all of these plots, in all 8 of these plots, all of the trees were tag measured mapped. We've done the stand reconstruction, we've done sap flow work, we've looked at the seed bank, seed dispersal, understory vegetation, animals, the adelgid coming in. We're really very opportunistic, right? It's like I give talks, I've been giving talks about this now for what feels like 15 years, and every time I say, you don't see yourself up here? Please come do it, right? And sometimes someone says, oh yeah, I'll come do mice, right? Or I'll come look at birds. You want to look at birds in our hemlock plots? I'd love that. We have a hard time keeping people interested in doing birds. And then we have permanent environmental monitoring stations in all of them. We look at nitrogen, we look at carbon. Because I'm interested in ants, we've done a lot of work with ants. But this is one of our girdled plots a year after the girdling was done, and you can see the light penetration through our ghost forest. Just an example to show you that, yeah, treatments do work. You kill trees and the canopy opens up. We've got here at the top, we've got controls for hemlock and hardwood. I take canopy photographs looking up with a fisheye lens twice a year, every year since 2003. And in the summer, the hemlock and the hardwood forests both have dark canopies, and in the winter, the hemlock and the hardwood forests are very different because the hardwood forests have lost all their leaves, and this is part of that ecosystem modulation that we saw earlier. Any day that the temperature goes over 32 degrees Fahrenheit, 0 degrees centigrade, those trees start photosynthesizing. They're active instantly, right? The girdle plots, the hemlocks just declined pretty rapidly. By 2007, we had almost 100% mortality. And you see similar things, both leaf on and leaf off. And when you log a plot and do a clear cut, you open up the sky. So the treatments really worked, right? No surprises there. And you can really see these changes in light penetration, just like we saw when the adelgid was out there. When the adelgid was out there in the woods today, the adelgid comes in, and even in the hardwood plots, the stands start brightening up as the few hemlocks in there disappear. There's a common color scheme that you'll see in the rest of the figures here. Blue are our hemlock controls. So when the adelgid gets in, they start brightening up both in the winter and in the summer. Yellow are our girdled plots, right? So you see the big jump after they're girdled. Red are the log plots, which are very bright. And purple are our hardwood controls. In terms of what these things are doing, in terms of just the basic what happens in the environment, so again, our same color scheme now. We've got girdle plots, log plots, and hardwood controls. These are differences from the hemlock controls. Right? So what this means is that the log plot in the summer is almost 2 degrees warmer than the control, and it's half a degree colder in the winter. So not only is there obvious temperature change, but the variance is much greater in both the valley and the ridge. And the variance is enormous in the soil. Just enormous in the soil. In the log plot, even in the girdled plots, you got a 3-degree change in the soil temperature in the summer and 1 to 2 degree change in the winter. So it's much, much hotter and much, much colder in a log plot. And over, in this case, the first 5 years of measurements, 6 years of measurements, the log plots and the girdled plots converge. So that slowdown, you get something really quick when you log it, but over time, this physical damage from the girdling does the same thing, and it's all doing the same thing as the hardwood controls, as the young hardwood. So in terms of their meteorology, they're doing very similar things. The vegetation is changing as well, and changing very dynamically. So in a paper that Elizabeth Farnsworth and I did back in 2012, we resampled the seed bank. The seed bank had been sampled first before we did the experiment. We sampled it again 6 years later, because the hypothesis is what's in the seed bank should come up, right? When you add more light to the forest floor, and just through general regeneration processes and mortality in the seed bank. So you have what's going on in the seed bank. We collect seed rain in those litter traps. So we've actually gone through all those litter traps, and we separate out all the seeds, and we identify all the seeds, and so we know what seeds are coming into these plots. We have vegetation, understory vegetation, looking at all of these. So again, we got the same color scheme. We've got our girdle plots, our log plots, our hemlock controls, and purple for our hardwood controls. And so you can see really, really big changes as we go, say, from 2004 to 2010. in our overall vegetation in the girdle plots as we go away from this side and towards that side in that particular thing for what's in the seed bank. So it loses a bunch of seeds. There's a lot of dynamics in the seed bank. There's even dynamics in the understory vegetation and the saplings. And if we pull this out, we can see that these forests are changing very, very dramatically. They're moving in direction. Some of the plots are moving towards what we think of as early successional species, more towards betula, particularly in the log plots in the first 6 years of this. We see the same thing if we look at what's going on in the overall forest floor vegetation, in the understory and the herb layer. There's not a big herb layer in a hemlock forest, but after a few years, you actually got quite a lot in that herb layer in the forest. You move into things that you might expect in mid-successional stands, including berberis, right? So this is one of the few places in Harvard Forest property where we've got berberis moving in. We get big changes in the insect fauna. I mentioned at the very beginning that we don't have a lot of ants, but as soon as you take the hemlock away, the numbers of ants increase, the number of beetles increase, the number of spiders increase. We shift. This is the girdle plot, one of the girdle plots just carpeted with spiders that on the forest floor. They're funnel web spiders that have just taken over controlling the fauna in the forest floor. In the log plots, we get tiger beetles, right? Because these need warm, these need open places. And so we get tiger beetles coming in, controlling the invertebrate fauna. We were particularly interested in how the ants intersected with the hemlock decline, because ants, I work at Harvard, right? Ed Wilson works at Harvard and says that ants are the little things that run the world, right? We all know that ants are the little things that run the world. And so the idea is that if you take hemlock away and the ants come up, you should have a whole new suite of ecosystem functions because you have all these ants running around there now. The challenge in this experiment is that how to disentangle taking what happens because of hemlock and what happens because of the ants, right? So because the two are very closely tied. You have a lot of hemlock, you have no ants. You take hemlock away, you have more ants. Is your effect on, say, soil nitrogen because you have more ants or because you took the hemlock away? Right? And so we actually did the experiment inside our big experiment. We made little plots, 3 meters on a side, where we kept all the ants away. Right? So we had ant exclosure plots. We used tangle trap and pitfall traps to get all the ants out. And so we had in our plots where you had no hemlock canopy, we also had plots with no ants. So we could compare the ecosystem processes going on with and without ants, with and without hemlock in the same plots. Right? So we've got our at the soil level, our controls with ants, disturbance controls, so we dug trenches, but we didn't put the tanglefoot on and we didn't put the barriers on, so we did all the disturbance, and then we kept the ants away. And we have this in our hardwood control, our girdle plots, our log plots, and our hemlock control and hardwood control. And then we watched this over a 7-8 year period from 2006 to 2014. We've got all our genera of ants. And the key message here is that at least for the first 8-10 years, the exclosures work pretty well. They definitely suppress the ants relative to the other ones. The maintenance kills you. By 2014, when we took the experiment down, there were just holes everywhere and things falling on our plots, and so it was like time to go. But at least for the first 6 years of this experiment, the exclosures work very effectively. And we actually saw pretty strong effects on this with and without the adelgid. So before the adelgid was there, in all of our different treatments, you have everything doing about the same thing, but after the adelgid, you start getting separation between the two, so that there's some interaction between the adelgid and the ants as well. And we can stuff this all into a structural equation model, into a path analysis model, to try and disentangle the effect of the canopy manipulation itself, the diversity of the ant manipulation itself, on downstream ecosystem processes such as decomposition, nitrogen availability, soil respiration, and the like. And the colored lines are the statistically significant effects. So we have strong effects of canopies themselves on ecosystem processes, and we have some effects of ant diversity on ecosystem processes. But the take-home message from this is it's really the foundation species much more than the ants that are running the world here. Now this could be because hemlock is really important. It could be because we just don't have as many ants in New England as we do in, say, Costa Rica. And so that's an interesting question as well. And so 20 years of experiments at this point have enabled us to say, yeah, this sort of conceptual model actually works pretty well, that we do have good control of these processes by hemlock itself, and we're actually convinced as a support for our hypothesis that it really does behave like a foundation species, and that when you take it away, you get new processes happening. So I'm going to finish up the last 5 minutes, and this is your cue for dessert if you want to set that out with where we're going in the future. And this is, we're going to dessert in the future. These are the volunteers for dessert. They needed to know when they were supposed to get up and put the cookies in. And I said, my last 5 slides are on the future. Right? So that was their cue. I want to emphasize, thinking back to our dispersal models, that these are models. All right? The models predict that it's going to get warmer and wetter in New England. And they predict particular trajectories. The models to date, every IPCC model prediction has been wrong. It's under-predicted what's actually happening. All right? So climate change is happening faster than the models predict. All right? But this is what the models have predicted. All right? And this is from the last IPCC report, not the one that was released last week. We know a bunch of other things. We know how the land was used, how agriculture happened. We know how we've moved from primary forest to secondary forest to agricultural land, and what this has done to our forest structure. We know what the soils are like. We know what our harvest rates are in New England, so we can put these things in as sort of state variables in the model. We know the diversity, the genetic diversity of the adelgid. Someone asked out in the woods today about the genetic variability in the adelgid. So these are AFLP lines just from Connecticut, Massachusetts, Pennsylvania, Virginia, and Maryland. And you can see the divergence here and the fact that things things show up in different ways. Massachusetts is showing up in multiple clades. So there's quite a lot of variability there. We know how fast hemlock is declining in our region. We can model the spread of the adelgid just using the climatic model and then adding in the human activity. And we can then talk about how quickly a patch dies after the adelgid gets in there. And so then we can run this out and we can say, okay, what's going to happen in terms of our vegetation and our biomass, key ecosystem processes, and the total primary productivity and heterotrophic respiration. And in these graphs, the dots are if the adelgid wasn't there, and the solid line is if the adelgid was there. Okay? And then we've got functional groups. We've got like early successional, mid-successional, late successional hardwoods. We've got pine thing, we have hemlock thing in this. So this is like in the model, this is a late successional evergreen. So this is hemlock, so it's going like this, and if the adelgid comes, it goes to 0. And then if the adelgid doesn't come, it keeps growing. And we did this for Harvard Forest, where the soils are good, except the soils suck. And then we did this for New Hampshire, where the soils really suck. strongly acidic poor soils. And we ran this simulation. We wrote this paper in 2000, and we published this paper in 2010 with Paul Moorcroft and Marco Albani at Harvard. And we projected this out and we said, you know, what would happen in terms of the key things that ecosystem scientists are interested in, net primary productivity, heterotrophic respiration. The adelgid would come in, we'd see a big drop that would be even bigger depending on how good or not good the soils are, and then it would slowly recover, and by 2100, maybe even 2050, we'd be back up at the same level of productivity. It's just now it would be birch and maple instead of hemlock driving this in the system. It would take much longer for respiration to recover in New Hampshire. And so this is for 2 sites, but we actually ran this for the entire eastern United States for the range of hemlock from 1995. So we could, the first 2 row and a half of this is data that we actually had when we ran the model. Right? And so red is when we have a loss of net ecosystem productivity. And then the blue is when it's more than it used to be. So that you can see spatially that as you move through time and as succession happens and as deciduous trees start coming in, and as the adelgid moves further west into the range of the hemlock, into the last range of the hemlock, and this process repeats itself as a wave, then you see you go from loss to starting to regain to it basically coming back in terms of net ecosystem productivity. And so if that's what you're interested in, it's like, okay, you lose one species, you gain another species, and you're good again. I will say that since we published that paper, we continue to collect data at our eddy covariance towers, and we have steady increase in net ecosystem productivity in our hardwood stands, and in our hemlock stands, though, in the green here, we see instead that it's moving its way. In this case, because these are atmospheric scientists, it's the other way, so they think of good or positive as going back into the atmosphere. And so what's happening is that this is becoming more of a source for carbon dioxide or carbon than it was in the past. And so now here we are in 2019, and we're right in this heavy red dot, right? And so 10 years out from publishing that paper, our eddy covariance data are supporting the way this model suggests it ought to go. Where does this leave us as we move into the future? You know, the forests are changing. I like to say, you know, our old forests are going away, and we're going to have a new forest. And as an ecologist, this is an extremely exciting and interesting thing to study. And it's been a real privilege to be able to study it here for 20 years and to really see how this is changing and playing out. At the same time, I really like hemlock, right? I'm really going to miss it, right? And I already am missing it. I walk through these forests, I walk through my experiment, it's a very different place. And how we as scientists, how we as people think about, deal with, and work with changing forests, whether it's because of land use changes or non-native species or climate change, is really the central issue that we're all confronting right now. Right? These systems are changing. They've always been changing. They're changing now. They're changing very rapidly. It's different in some ways for us to sit and look at this and say, wow, this is really changing fast, and I had a part in that change. But they do always change, and we need to think about that too, and think about how that works. So thanks very much. We've got dessert, I'm sure, and we can chat and talk, take questions, or however you'd like after that. Thanks very much. Absolutely.
Are there any specialized species that your work or others have identified that you feel like maybe sort of dragged down with the loss of hemlock, so amongst the invertebrates or other groups?
Well, I mean, you know, I'm not much of a birder, but there are a handful of warblers that are not doing very well with that. There is a whole suite. There are about 300 species of hemlock canopy specialist spiders. They'll all go, right? They're only in hemlock canopies. They're toast, right? I mean, you know, I put together the field guide to the ants of New England, and, you know, well, we got 130 species. It's just like the ferns, right? You can do ferns, you can do ants. They're like, they're the only regional similar book we have for spiders is the Spiders of Connecticut, which is about 20 years old now. It's 1,600 species of spiders in Connecticut. It's just vast. So there's spiders. There's a whole group of fungi that are specialists on hemlock and on other softwoods, but really focus on hemlock. I don't actually know a whole lot about fungi and soil fungi, but I know enough to know that when you switch from a hemlock-dominated forest to a hardwood-dominated forest, you switch from, I hope I don't get it backwards, brown rot fungi to white rot fungi, or the other way around. But one of them, the ones that eat hemlock are very fast in terms of turning over the decomposition, and the other ones that do hardwoods are very slow. So your whole rate of soil decomposition and soil turnover and microbial activity changes by 2 orders of magnitude when you shift from hemlock-dominated forest to hardwood-dominated stands. So we're going to have to watch that, and we have groups interested in this, for 100 years to really understand how that's going to play out in our soils. But we're looking at a rate change on scales of centuries for the fungal turnover.
So you had mentioned that foresters are saying remove the hemlocks, and I've been in courses recently where they say, no, we made a big mistake, we didn't leave like the new chestnuts because there might have been some that were
Resistance, exactly.
So, I mean, do we really never learn from the past? Is there really no one keeping some hemlocks that might?
So there are. There's a group at the University of Rhode Island, Evan Pricer's group there. A graduate student of his, Laura Ingwell, about 10 years ago now, worked with URI and Extension to put together a flyer that basically says, have you seen this hemlock? It's got a picture of a green hemlock surrounded by dead hemlocks, right? And they printed up, I don't know, 100,000 of these, 1 million of these leaflets, and they sent them all over the East Coast. And anytime someone would pick one up, say, oh yeah, I saw one up on the hill. And they'd send an email to Laura, and Laura and Evan would go, and they'd get some cuttings or stock, and they'd bring it to URI. And they've been working on this, and they've been working on identifying lines that are less susceptible to the intelligent. There are also groups that have taken stock and have planted it in orchards in Patagonia. And they're waiting. So once we deal with the intelligent, or it goes through and burns itself out, we can replant it. Will we be able to replant 6 million square kilometers of hemlock? Can we do that with chestnut? It's not stopping the American Chestnut Foundation from trying. The question of, do we never learn? I can't answer that. I mean, we're doing it again with ash. What can I tell you? The goal, the desire for money outweighs the desire for common sense. And to be fair, we all care deeply. Ecologists care deeply. There aren't very many of us.
So do you see another foundation species, you know, after, I mean, you have the chestnut.
Right now, Right now, no. You know, what the mixed hardwoods are basically, even chestnut to some extent, are basically swappable. Chestnut is an interesting case. We, unfortunately, better or worse, this was 07, 17, we don't have the data. We don't know what was, you know, we know, of course, animals needed the chestnut mass. We don't know what happened. We don't know. We know that chestnut leaves don't, for example, decompose much more differently from oaks or beech in leaf packs. But right now, it's curious here that, you know, one of the main species that people look at as a foundation species in Colorado in the west is whitebark pine, and those two are in trouble. But on the other hand, some of the other ones in the southwest are moving north with climate change. So in the west, it looks, in some ways, it's more interesting in that way. Here, I don't know yet. I don't know yet. And one of the challenges in a whole nother line of research is we use experiments like this in natural occurrences like hemlock decline, like whitebark decline, like coral reef decline, to say, oh yeah, that species was really important. It would be really nice to be able to go in and say, you know, this species, it's super duper important. I know it's common as dirt. I know the Endangered Species Act doesn't pay any attention to it, but really pay attention. And as an example of that, okay, white ash, their current estimate is that there are 8 billion white ash trees that are alive. IUCN red listed it as globally endangered this year. Right? It's not, the U.S. Endangered Species Act isn't going to deal with it, but IUCN listed it this year. Right? Now, that's different. You know, that's not 1,800 pandas. That's 8 billion ash trees globally endangered today. Right? That's a big step. Right? But I got a whole nother sort of line of research now, 20 years on, trying to use clever statistics and these large forest dynamics plots to identify at least interesting species, you know, candidate species that deserve more look. But you still need 20, 30 years of experiments to convince me, right? I mean, I'm a scientist. I'm pretty skeptical about these things, so it's hard.
What about Carolina hemlock? First, is it susceptible to endangerment?
It is, and it's endemic. Yeah. I honestly don't know. I mean, it's so patchy, and when I've been down, it's a number of years ago now, when I was down at Coweta Experiment Forest in wherever it is, Georgia, South Carolina, it's right on the border, so it's always puzzling to me. I don't think Carolina hemlock is doing especially well. But it also has been outplanted in Patagonia. So there's some, I forget the group, but they're grabbing all these northern hemisphere trees that are under threat, and they're planting them somewhere else. Because we group working here and at Cary Institute for Ecosystem Studies in New York a couple years ago now did a review and report that was both popular press as well as the peer-reviewed things on pressure of non-native insects and pests and pathogens. The Northeast U.S. is ground zero, man. We got 57 non-native insects, fungi, pathogen in or just about to be at outbreak on our native forest trees. More than anywhere else in the world. Right here. It's like everything. Janet.
A lot of the examples that you gave of things breaking down besides the foundation species were sort of closely about co-evolution things. And I was wondering, I mean, there has always been change, and there, you know, this is not unusual, except for the frequency of change.
The rate.
The
yeah.
rate of change now is so much because we're moving things around so much more. And it seems to me like that would affect the possibilities of coevolution. And which is kind of a real evolutionary switch.
Yeah, I think so.
And I wonder what effect that.
I don't know. I honestly don't know. I think it's a really interesting question, and we could spend like 20 years on it. But one of the most interesting, in a lot of ways, and the Adelgid is reflective of this as well, but one of the most sort of interesting avenues of the reintegration of ecology and evolution is that evolutionary, because so when I was in graduate school, and I walked up and hill both directions through the snow to go to school, but we used to have biology departments and biology PhDs, and someone studying ecology would take evolution and vice versa, and that's not the case anymore. You have departments of ecology and departments of evolution. You can take, and lots of my younger colleagues have ecology degrees and they don't essentially know what natural selection is. And vice versa, right? I have a lot of colleagues in evolution who don't know what Lotka-Volterra competition equations are, right? But both sides are rediscovering that, wow, evolution can happen in ecological time. And so we can really see rapid evolutionary change, gene frequency change, in the time scales that ecologists do their PhDs on. And so this is surprising evolutionary biologists, and it's causing ecologists to look at evolution more, but it allows us to hone in more on rapid coevolutionary dynamics. I don't know if anyone's doing it, but it's possible to do that now. We have the methods to do that now. So it'd be really cool to look at. But I don't know, one of the, we see rapid genetic and evolutionary change in the Adelgid, even though it's an entire asexual lineage. What does this mean for something like resistance with the evolution, the rapid evolutionary resistance or acquisition of resistance by hemlocks? I don't know, right? If we weren't cutting it down so fast, we'd have more material to study it.
If it's anything like crops, because they're, you've got crops that are, and they just have to, you have to rotate those lines constantly.
Constantly, because it's very, very quick.
Yeah.
It's very, very quick. So.
I wonder, oh, sorry. Go ahead.
Thinking about what you were saying about the fungal floral changes that might be happening through time, decline of hemlock. What's been done here at Harvard Forest in your plot areas, especially in terms of baseline information about the fungal composition?
So Sarita Fry and her group at the University of New Hampshire have been focused on the changes in bacterial and fungal populations and genotypes in the hemlock plots and hardwood plots, and we just amass immense amounts of information about that. I can't tell you where all of it is in the pipeline.
Okay, so at least it's a baseline out there.
Oh, yeah,
they're useful down the road.
yes,
Okay,
absolutely. And it's paralleled. We have, it's all another universe, right? But we have, with the LTER, we have a number of climatic warming experiments here. So we do soil warming, air warming experiments, and we also look at all these changes in those experiments as well. So we, even though they're done in different places, we put all these pieces together in different ways.
I guess I was just going to follow up on the question you asked about this idea that some of this change is just, maybe it's just accelerated compared to the past, but it's not that different than the past.
Well, my point was maybe it is different.
Yeah, so I was just wondering if it's almost like maybe this is more analogous to like a biotic turnover event where you're, introducing two biotas to each other. Basically East Asia is now has access to Eastern North America, which is sort of this evolutionary mismatch between those two regions.
No, it's a very exciting time to be an ecologist. Thanks very much. I have two announcements before we stop. The first, and there's a flyer about this out on the table next to dessert. Next weekend, next Saturday from 10 to 4, we're having the official closing of the Hemlock Hospice exhibition. It's actually going to be up through mid-November. And then we're supposed to take it down, although we don't know really what to do with it. If anyone wants any of the pieces or has any ideas, please talk to me. I mean, we'd be happy to put it mounted somewhere else. But we're going to have a day of events. We're going to have tours, walkthroughs of it from 10 to 12 and 2 to 4. And in the middle, from noon to 2, we're going to have a panel of presentations and discussions by 3 folks who work on non-native species. We're going to have Laura Meyerson up from the University of Rhode Island, Val Pasquarella from Boston University, who's working on gypsy moth, which has been an outbreak in our region right now. Dave Orwig, who works here at Harvard Forest, talking more about the adelgid. David Foster, our director here at Harvard Forest, doing some of this in context. What, free lunch, blah, blah, blah. There's a poster out there. Please, pass it around, bring your friends. We'd love to have people show up. As I said, if there's, if any of you has any ideas, would like this in their piece in their backyard, please talk to us. We are putting it, we have a spin-off piece about climate change and warming. That's a really big sculpture by my universe. It's 30 feet by 11 feet by 11 feet that we're installing on Harvard's main campus on Monday. It'll be up for 8 weeks as well on the Science Center Plaza. And for those of you who are, really keen on learning more, I have 4 different books out there. My Carnivorous Plant book, the Field Guide to the Ants of New England, our Harvard Forest book on hemlocks, and a book of photography from the Northwest. If anyone's interested in buying and having one signed, we got special NEBC deals tonight. So talk to me over dessert.
So I want to
Thank you very much.
Thank you so much.